Okay, in chapter 26, we're going to talk about normal renal function. So with the kidneys, we have two of them. They're sort of bean-shaped organs that sit in the posterior abdominal wall, and the main functions of the kidneys are to help maintain fluid and electrolyte homeostasis, as well as helping to rid the body of any sort of water-soluble wastes. Now, what's interesting is the kidneys can filter more than 7 liters of fluid per hour. This is more than your entire blood volume on average. And of that 7 liters that's filtered per hour, most of that's reabsorbed. 99% of that or more is reabsorbed. And so then what the kidneys can do then is produce a small amount of urine that's highly concentrated in wastes. That way we can hold on to a majority of the volume of that fluid and electrolytes, but then deliberately help eliminate some of the wastes from blood. Now, looking at a view of the abdominal cavity, what we see here is we got two kidneys on either side, just inferior to the liver and posterior to structures like the stomach, pancreas, and other intestines. Now, each kidney is served by a renal artery, which comes off of the aorta, abdominal aorta, and then is drained by a renal vein, which then goes into the inferior vena cava here. Now, the kidneys have a muscular tube that connects down towards the urinary bladder called the ureter. So each kidney has its own ureter that basically carries urine down towards the urinary bladder, and the urinary bladder can store that urine until micturition or voiding or elimination of that urine. Now, looking inside of the kidney, this is still gross anatomy. What we can see here is an outer layer of cortex. So the renal cortex is the outermost layer. Cortex means bark, so it's the outer layer, kind of like bark on the tree. Deep to that, we have a medulla, and the medulla has some different structures in here. Now, each kidney is surrounded by a renal capsule on the very outside, which is kind of like a fibrous tissue that encases the kidneys and helps support and protect each kidney. Now, within the cortex, you're going to find a lot of glomeruli. So the cortex has an abundant amount of glomeruli, which we'll talk about here in a minute. And these are basically the filtering mechanisms of the kidneys you find within the cortex. Now, other extensions of the microscopic filtering mechanisms, called nephrons, then dive down deeper into the medulla, where we have aggregations of nephrons here at what we call the renal pyramids. So the renal pyramids are basically just large collections of nephrons that all converge at what we call a renal papilla. So the renal papilla is the very tip of the renal pyramid right here, and it's at this renal papilla that highly concentrated filtrate is then drained into these spaces called the minor calyces. So here's a minor calyx and there's a minor calyx, and this is basically where urine is first formed, or sort of called urine, because once that fluid or urine enters the minor calyx, it's no longer modified any further. Like, you're not going to reabsorb any of those substances. You're not going to secrete any additional substances into that fluid you're finding within the minor calyces. Now, each minor calyx will come together to form a major calyx, and then when all the major calyces come together, they form the renal pelvis. The renal pelvis is just a large sort of space here, deep within the kidney itself, and that leads into the basically the hilum, which is the place where all the vessels either enter or exit the kidney. And the renal pelvis leads into the ureter, which is a muscular tube that can help transport that urine down towards the urinary bladder. Now, we know the kidneys receive a lot of blood flow. If they're filtering out 7 liters of fluid every hour, this means then that they're receiving a pretty significant proportion of cardiac output. And so the kidneys receive blood flow from a renal artery, which comes off the abdominal aorta. This renal artery will then branch into the segmental arteries here, and then those go into the interlobular arteries, which then wrap around to form an arcuate artery. So these arcuate arteries kind of wrap around the boundary between the cortex and medulla. Now, coming off the arcuate arteries, what we find then are the cortical radiate arteries, and these cortical radiate arteries will project up into the cortex, and they have branches coming off, which we have the efferent arteriole, or afferent arteriole, which serves the glomerulus of the nephron. Now, we can't see those because they're microscopic, and each kidney contains about 1 million nephrons. You're going to find about 1 million nephrons within this one kidney here. Now, eventually what happens is that those blood vessels will reconverge into the veins, so then you have arcuate vein, interlobular vein, segmental vein, renal vein, and then back into the inferior vena cava, and so back into the systemic venous system. So if we zoom in on what a nephron looks like, it's basically what we see here. And the nephron is a system of tubules that basically help pick up filtrate from the glomerulus, and all along the way that filtrate gets modified from the proximal convoluted tubule down through the descending limb of the nephron loop, the ascending limb of the nephron loop, to the distal convoluted tubule, and then finally the collecting duct. Now, it all starts here at the glomerulus, and the glomerulus is basically an aggregation of glomerular capillaries as well as Bowman's capsule, or the renal capsule, which surrounds those glomerular capillaries. So what happens is that the capillary hydrostatic pressure forces fluid out through the glomerulus, it crosses Bowman's capsule, and it enters this capsular space here. In fact, once that fluid enters the capsular space, we call that filtrate because it was actually filtered from the glomerular capillaries. Then that filtrate flows throughout the nephron where it's modified. Now we know that more than 99% of that filtrate gets reabsorbed until eventually we get towards the collecting duct where we're going to find a smaller volume of more concentrated urine. Now, you'll notice that this nephron here, the whole system of tubules, it's also associated with a lot of blood vessels. So the purpose of these blood vessels is to help reabsorb some of the substances from the nephron, as well as secrete substances into the nephron. So different wastes and drugs, things can be deliberately secreted into the nephron, which can enhance their excretion or elimination from your body. So if we just look at the nephron by itself, what we see here then is we have a little afferent arteriole leading into the glomerulus, and then an efferent arteriole carrying blood away from that glomerulus. And what happens then is that due to capillary hydrostatic pressure, fluid gets forced across this capillary membrane, across Bowman's capsule, and then into the capsular space. And then due to pressure, what happens is that fluid then gets forced along the length of the nephron. We have the proximal convoluted tubule, which is first. That leads to the nephron loop, or loop of Henle, which then leads into the distal convoluted tubule, and then the collecting duct. Now, most reabsorption, or basically movement of substances back into your bloodstream occurs here in the proximal convoluted tubule. So about 90% or more of substances get reabsorbed right away from the proximal convoluted tubule back into the bloodstream. Now, there's some additional processing that occurs in the nephron loop, and the distal convoluted tubule and collecting duct are under the control of different hormones that we'll talk about here in a minute. So if we zoom in on the structure of our glomerulus here, what we'll find is these glomerular capillaries are, as they sound, they're capillaries, which are fenestrated. So it means they have lots of little holes in them. Now, these fenestrations improve the leakiness of these capillaries. So it means that these capillaries can help leak fluid out into the capsular space due to things like capillary hydrostatic pressure. But what also helps maintain the leakiness of these glomerular capillaries are these things called podocytes. And these podocytes have these little processes that stick out called pedicles, which basically means like little foot or feet. And these pedicles interlock to form what we call filtration slits. And these filtration slits help enhance the selective permeability of this membrane. That way larger proteins can't just cross through the fenestrations of this capillary. So what we find then is that what gets filtered across the glomerular capillaries then are going to be smaller ions and water and wastes, but larger things like cells and large proteins aren't allowed to normally cross this capillary membrane here, at least in the glomerular capillaries. Now, this yellow substance here you find in the capsular space, well, this is filtrate. And filtrate is what's basically been filtered from the glomerular capillaries that gets forced into the proximal convoluted tubule, where then many of those substances can get reabsorbed back in the bloodstream by things like active transport or facilitated diffusion. So in the proximal convoluted tubule, just after the glomerulus, this reabsorbs about 2/3 of the filtered water, electrolytes, and glucose, as well as amino acids, proteins, and vitamins. So a majority of that filtrate gets reabsorbed almost immediately. Water is reabsorbed passively due to osmosis, because once you start using ATP to reabsorb things like amino acids, proteins, glucose, and vitamins, well, you're basically moving solutes back into the bloodstream, so then water follows those solutes through osmosis, which is a passive process. So what we find here then is that filtrate moves down through the tubule system. We can use ATP to help reabsorb things like glucose, amino acids, and chloride. Water will follow due to osmosis. And this occurs at the apical membrane and the basal membrane, where then those substances can get reabsorbed back into your bloodstream. That way you don't lose these important nutrients. Things like glucose, amino acids, and water, as well as sodium, potassium. These are important nutrients for our body, and you wouldn't want to necessarily always lose these things in urine. There may be some circumstances where you want to get rid of excess glucose, sodium, or potassium, but it turns out we have mechanisms to help eliminate those particular substances. So for the most part, though, these are reabsorbed immediately in the proximal convoluted tubule. Now, what you find in the proximal convoluted tubule too are a lot of these microvilli, and these microvilli here help increase the surface area for absorption, and that's why the absorptive rate is really fast in the proximal convoluted tubule. Now, what occurs in the nephron loop is what we find is that the nephron loop has a descending limb and an ascending limb. And in the descending limb, it's only permeable to water. And in the ascending limb, it's permeable to things like sodium, chloride, and potassium. So what happens is that while filtrate is flowing up through the ascending nephron loop, we actually have these sodium, chloride, and potassium pumps that use ATP to pump out sodium and chloride from filtrate back into the interstitial fluid of your kidneys here. So basically you're reabsorbing sodium and chloride from filtrate into the interstitial fluid of your kidneys, and this sets up an osmotic gradient. In fact, if you look here in the medulla of your kidney, you'll notice that all these little dots represent the number of solutes, and you'll find that as you get deeper and deeper in the medulla, the solute concentration increases. And the reason why you have a high concentration of solutes here in the medulla is that we actually use ATP to pump out sodium and chloride into that medulla, and that sets up an osmotic gradient, which allows you to reabsorb water from the descending nephron loop. So what we find then is that water gets reabsorbed in the descending nephron loop, which makes it a smaller volume of more highly concentrated filtrate. And then as that filtrate moves back up to ascending limb, those solutes then are actually also pumped out. So what you end up with then is a smaller volume of also more dilute filtrate by the time that filtrate gets to the distal convoluted tubule. Now, once at the distal convoluted tubule, the filtrate can be modified further, as well as in the collecting duct here. So many of these substances get reabsorbed back in our bloodstream, and this helps maintain the osmotic gradient here, which allows you to reabsorb things like water, sodium, and chloride. Now, in the distal convoluted tubule, the filtrate is hypoosmotic. We reabsorb so much solutes and some water as well, that it actually made it hypoosmotic. So it had less osmolarity than you would find in blood. And further reabsorption of things like water and solutes is mediated by hormones like aldosterone, angiotensin II, the natriuretic peptides, or urodilatin. And we know that aldosterone helps increase sodium reabsorption. This is one of those hormones that helps increase blood pressure, because it's released as part of the RAAS, or the renin-angiotensin-aldosterone system. And aldosterone, it's released from the adrenal cortex, but it acts on the distal convoluted tubule to help reabsorb sodium. And we've learned earlier that if you reabsorb sodium, then you also reabsorb water, because water follows those solutes. So that by reabsorbing sodium, you can also reabsorb water and help retain that water. This can help increase blood pressure by increasing blood volume. Now, angiotensin II also plays a role in stimulating sodium and water reabsorption in the kidneys. Now, the natriuretic peptides and urodilatin, those actually inhibit reabsorption. So we've learned that the natriuretic peptides, or atriopeptin, is released from the heart, or the atria, when the atria were excessively stretched, and you'd assume that this occurs with high blood pressure. And so these hormones are released to inhibit sodium reabsorption, which then also prevents the reabsorption of water to help decrease blood pressure. So you can see right here that these hormones act on the distal convoluted tubule to alter how filtrate is formed, to also influence things like blood pressure and electrolyte homeostasis. Now in the collecting duct, this is actually where a lot of distal convoluted tubules will converge, and the collecting duct is sensitive to antidiuretic hormone. So antidiuretic hormone, or ADH, also called vasopressin, is released by your posterior pituitary gland, and it can be released in response to things like low blood pressure or high solute concentration in your bloodstream due to dehydration. And what antidiuretic hormone does is that antidiuretic means it prevents diuresis because it's antidiuretic. So it prevents diuresis or water loss. So antidiuretic hormone increases water reabsorption at the level of the collecting duct. That way you don't lose as much water in urine. And so the urine that's created in response to antidiuretic hormone is a smaller volume of highly concentrated urine. Now, the kidneys also have something called glomerular filtration rate, or GFR. Now, glomerular filtration rate is determined by things like filtration pressure in the glomeruli, as well as the permeability of those glomeruli. And a normal GFR is about 125 mils per minute. Now, if GFR is too fast or is too slow, that can indicate some pathologies that may be present. Now, the way that glomerular filtration rate is regulated is through intrinsic and extrinsic mechanisms. One of the intrinsic mechanisms just relates to blood volume. So we know that if blood volume increases, blood pressure increases, and if blood pressure increases, hydrostatic pressure increases, which actually causes more fluid to be forced out of the glomerular capillaries more quickly. So we say that due to increases in blood volume, this is actually going to increase glomerular filtration rate, which also increases the amount of fluid that's excreted, or the amount of urine that's formed in response to that. This is one of the ways that blood pressure and blood volume are regulated by the kidneys. Now, opposite to this, if your blood volume is too low, then your blood pressure would presumably also be low, and that would actually decrease glomerular filtration rate because of a decrease in hydrostatic pressure of the capillary, so that fluid is actually conserved as a result of this decrease in GFR. And these are just part of the autoregulatory mechanisms here. Now, if you get wide fluctuations in glomerular filtration rate, there are other mechanisms that can help regulate GFR through increasing or decreasing blood flow to the glomerulus, especially under conditions of hypertension, where you might expect that if someone's blood pressure is too high, it could potentially damage the glomerular capillaries. And so there's autoregulatory mechanisms that can diminish blood flow to the kidneys if blood pressure is too high. It helps protect those glomerular capillaries from potential damage. Now, one of the intrinsic mechanisms is this tubuloglomerular feedback. And with tubuloglomerular feedback, each individual nephron can regulate its own glomerular filtration rate. This is done through a mechanism where essentially the glomerulus can be associated with a structure called the Macula Densa, and Macula Densa measures sodium content in the filtrate. And we'll learn here in a minute how sodium content in the filtrate gives us a sense of how fast or slow GFR is. So it involves the glomerulus, macula densa, and these juxtaglomerular cells that can release a hormone called renin. And so what we see here is that in this juxtaglomerular apparatus, which you find near the association of the glomerulus and distal convoluted tubule, what we find then is that the distal convoluted tubule comes back near the glomerulus. And so remember, the distal convoluted tubule is actually going to be near the end of the nephron. And so what's neat here is that the end of the nephron will associate with the beginning of the nephron, and it's a way to help sort of check how filtrate was actually modified once it gets to the end of the nephron in comparison to the beginning. So the cells that monitor filtrate here at the distal convoluted tubule are called macula densa cells. And these macula densa cells sense the concentration of sodium and chloride in the filtrate. And once they regulate, or once they can sense different concentrations, they can communicate with these juxtaglomerular cells. And these juxtaglomerular cells can actually help regulate the diameter of the blood vessels that serve the glomerulus. So we know then that if these blood vessels are bringing in, blood to the glomerulus itself, if we vasoconstrict this afferent arteriole, you're going to decrease blood flow to the glomerulus, which would decrease blood pressure in the glomerulus and therefore also decrease glomerular filtration rate. And the opposite would be if you vasodilate this afferent arteriole, you can increase blood flow to the glomerulus, which can increase blood pressure here, increase glomerular filtration rate as a result. So how does that relate to this whole system here? Well, in the distal convoluted tubule, if the concentration of sodium chloride in the filtrate is too high, what this suggests then is glomerular filtration rate is too fast. Because if GFR is too fast, then there's not enough time to reabsorb those solutes back in your bloodstream, which means by the time that filtrate gets to the distal convoluted tubule, you're going to have a lot of solutes left behind because they haven't had enough time to be reabsorbed. So the macula densa here can sense that, and once it senses that sodium chloride concentrations are too high in the filtrate, it can cause the afferent arteriole to vasoconstrict, which can decrease blood pressure in the glomerular capillaries, which decreases glomerular filtration rate. Opposite to that, if GFR is too slow, what we find then is that the macula densa cells here can sense that sodium chloride is too low in the filtrate. And you can imagine that if GFR is really slow, that filtrate is moving very slowly through the nephron and therefore has a lot of time to be solutes have a lot of time to be reabsorbed. So that the solute concentration in the distal convoluted tubule actually would be very low. And once the macula densa cells sense that solute concentration is low, they'll cause vasodilation of the afferent arteriole, which then increases blood flow to the glomerulus, which increases GFR and helps keep GFR at a steady level. And this is how glomerular filtration rate is regulated at 125 mils per minute at each individual nephron. Remember, there's millions of nephrons in each kidney. So it's pretty neat that each individual nephron is more finely tuned through this juxtaglomerular apparatus. Now, we mentioned earlier how these macula densa cells can sample sodium chloride content in the filtrate. And if in the distal convoluted tubule, what we find that if sodium chloride concentrations are too low in the distal convoluted tubule, this will lead to a compensatory mechanism that increases GFR. And if sodium chloride concentrations are too high in the distal convoluted tubule, this will lead to a compensatory mechanism that decreases GFR. And so we can use the concentration of sodium chloride as an indicator of how fast filtrate is flowing through the nephron. Now, we know the kidneys also regulate blood volume and osmolality, and so it can actually alter osmolality by altering GFR, which can alter the reabsorption time from filtrate. So the way this occurs then is our kidney tubules are responsive to various hormones that are used to regulate osmolality, or basically electrolyte balance in our bloodstream. So the first one we'll talk about is antidiuretic hormone, or ADH, also called vasopressin. Now, antidiuretic hormones released in response to situations like low blood pressure, blood volume, or high solute content in the blood. And so what antidiuretic hormone does to the kidneys is it increases the permeability of the collecting tubule to water. And by increasing this permeability to water, it can increase in the reabsorption of water, which can actually help reduce blood osmolality, or basically help reduce the concentration of solutes in blood and prevent like hyperconcentration of solutes or hypertonicity of your bloodstream and body tissues. And so the way this works then is that ADH, it's released by your posterior pituitary gland, binds to receptors on cells here in the collecting duct of your kidneys. This leads to an intracellular signaling cascade that leads to the inclusion here of what we call aquaporin proteins in the collecting tubule lumen. And by putting these aquaporin proteins here, we can actually help reabsorb water from filtrate. So in response to antidiuretic hormone, what we end up with then is filtrate that's more highly concentrated and a smaller volume due to the reabsorption of water. So that the urine you're going to see in response to ADH is going to be in lower volume and more highly concentrated. So it's going to look darker in appearance. Now, there are some problems that can occur due to antidiuretic hormone. Insufficiency of antidiuretic hormone could come secondary to things like pituitary damage, and this can result in a condition called diabetes insipidus, where basically you aren't releasing enough, antidiuretic hormones. So you see excessive water loss through urine. So you see large volumes of dilute urine that are excreted, and this can lead to pretty severe fluid imbalances. Or we might find that the collecting tubules are no longer responsive to antidiuretic hormone. We call this nephrogenic diabetes insipidus, where you might have enough of the hormone present, but if the tubules are no longer responsive, then you can't reabsorb water in response to that hormone. So then you still end up with excessive urination and water loss which again leads to fluid and electrolyte imbalances. Now, diuretic agents are drugs that alter the osmolality of urinary filtrate, and they can help oppose the reabsorption of things like water. That's why it's called a diuretic agent, is that diuretics increase water loss, so it increases the urine volume. Now, osmotic diuretics will increase the osmolality of filtrate, which causes more water to remain in the tubule, and therefore water is going to be more likely to be excreted or lost in urine. And there's different types of diuretic agents. ACE inhibitors, which block the formation of angiotensin II and aldosterone, would also basically act on that osmolality system we talked about, because if you prevent the secretion of aldosterone, then you're not going to reabsorb as much sodium, which means sodium stays in the filtrate, and because water follows solutes, then you're going to lose more water in filtrate and in urine due to the inhibition of aldosterone. Otherwise, things like loop diuretics can block the reabsorption of different electrolytes like sodium, potassium, and chloride, and those can also block this reabsorption in the ascending loop, which then also increases the water loss, which is why it's a diuretic. As well as these thiazide-like diuretics can block sodium reabsorption, along the nephron, which again increases water loss because water follows solutes. Because you're losing those solutes in filtrate by not reabsorbing them, then you're going to increase water loss in urine. Now, the kidneys actually can release hormones themselves. So what's interesting is the kidneys normally release a hormone called erythropoietin, which is actually a growth factor for red blood cells, as well as they serve as in the activation of vitamin D3 or calcitriol. And this is a necessary cofactor for calcium reabsorption in the intestines. So in conditions of chronic renal failure, what we're going to find then is that when the kidneys start failing, then they stop releasing erythropoietin. Well, then you can't stimulate red blood cells to grow, and this can result in anemia or a deficiency in oxygen transport. Or you can also see if the kidneys are failing, then you have less vitamin D activation, and this leads to calcium or hypocalcemia, or low blood calcium due to vitamin D inhibition. Now, we can test normal renal function through a variety of mechanisms, and we can do this in either urine or blood. And so some of the most helpful tools in evaluating kidney functions include things like urinalysis, where you just directly look at urine itself. We can look at serum creatinine, blood, urea, nitrogen, and these are just both blood markers of kidney functions, or other tests of glomerular filtration rate. Now, otherwise, we can look at kidney structure through using other diagnostic tests like ultrasound. Now, in urinalysis, what we can do then is actually, basically measure the content of urine to get an idea about normal kidney function, or if it's abnormal. And there's different types. We can look at single sample or 24-hour types of urinalysis. In a single sample, you just get one sample of urine and you look at its contents to see if the kidneys are functioning normally, and you're finding, normal concentrations of wastes, or you might not want to find things like protein or cells in urine. If you are, then that can suggest kidney damage. In 24-hour samples, we find that is that you can actually inject a known quantity of a substance in someone's bloodstream, and then 24 hours later, basically measure how much of that substance you find in their urine. And this can give you an idea of how well the kidneys are able to clear out that substance from the bloodstream. We can look at culture and sensitivities to determine the presence of things like microorganisms to figure out if someone has an infection. Now, we can also look at odor. We know that normally urine has sort of a slight sort of, urea or nitrogenous sort of odor to it. But if urine is very strongly smelling of ammonia, this can indicate things like infection, bacteria. Otherwise, food can cause different odors too. So this food can be a confounding variable that can complicate this form of urinalysis. Normally the color of urine should be sort of pale yellow and amber due to urochrome pigments, which come from bilirubin. Now, otherwise, if it's brown or bright red, this can indicate hematuria, which is blood cells in urine. If it's cloudy, this can indicate infection. Otherwise, if it's dark yellow or orange, this could be highly concentrated urine, which may also indicate a decrease in things like GFR. Otherwise, there are different drugs and foods that can alter urine color, so this is something to keep in mind. Now, there are genetic factors that can influence urine color too, like alkaptonuria, which is a black urine disease, And with this particular genetic condition, these individuals have difficulty processing a particular type of amino acid, and in result, this amino acid builds up in their bloodstream and it ends up looking like a darker pigment in urine. So their urine ends up looking kind of more black. Now, normally urine is kind of clear, pale yellow, amber, and slightly acidic. Any kind of abnormalities, things like excess protein, red blood cells, crystals, any sort of stones or casts can indicate, infection or disease along the urinary tract or in the kidneys themselves. Urine osmolality and specific gravity should normally vary over the course of a day due to fluid intake, but a fixed osmolality or specific gravity can indicate renal impairment, where if you're not seeing normal fluctuations in GFR due to increases or decreases in blood volume from fluid intake or fluid loss, this can indicate kidney disease. Now, in urinalysis, what we find is that there are some abnormal substances that you shouldn't normally find in urine. You shouldn't find a whole lot of protein in urine. A small amount is normal, but if there's an excessive amount of protein in urine, we call this proteinuria. You shouldn't find a lot of glucose in urine, because normally you should reabsorb a lot of sugar in your nephrons instead of losing that in urine, because this is an important nutrient. If you're finding it in urine, we call this glycosuria, which is like a sweet urine. In excess epithelial cells, red blood cells, leukocytes, or bacteria can indicate infection or disease. Now, crystals and stones can also indicate disease, and there's different types of casts that these crystals and stones can create. We can have white blood cell casts, which are associated with renal infections, which we call pyelonephritis. Red blood cell casts indicate infection of the glomerulus and damage of the glomerulus, which we call glomerulonephritis. Otherwise, epithelial cell casts can indicate sloughing of tubular cells, which can be due to like autoimmune disease or infection, which we call acute tubular necrosis. Now, one of the serum markers for normal kidney function is serum creatinine, as well as blood urea nitrogen. Now, normally creatinine and blood urea nitrogen are things that should be eliminated by your kidneys. If you're finding an excessive amount of creatinine or blood urea nitrogen within the bloodstream or serum, this indicates a decrease in GFR, and we can use this as a way to sort of indirectly measure kidney function. So serum creatinine is more reliable than blood urea nitrogen, because blood urea nitrogen is actually altered by things like protein intake. If someone has a diet that's high in protein, that's going to increase their blood urea nitrogen, which could indicate that their kidneys are damaged or failing, but it's not due to that. It's actually due to their particular diet. Creatinine is also a more reliable indicator of renal function, but you can look at both as a measure of GFR. Now, creatinine levels are affected by two different factors. We know that creatinine is produced from muscle, so that if someone has a lot of muscle activity, this can actually increase creatinine levels. And the rate of creatinine excretion by the kidney is actually determined by GFR. So if someone has really high creatinine levels in their bloodstream, this indicates their GFR is actually low, and elevated levels of creatinine then therefore suggest a decrease in GFR. Now, blood urea nitrogen is a byproduct of protein metabolism, and normally you should find it within 10 to 20 mg per deciliter. And if it's elevated, this can indicate a decrease in renal function, a decrease in fluid volume, or an increase in dietary protein intake. And this is why blood urea nitrogen is not a super reliable indicator of kidney function, because it's altered by dietary protein intake, but it's something to consider when you're evaluating someone's kidney function.